Let's welcome Dr. Bernadette and Dr. Mishra.
Thank you for the introduction. So I'm Neha Mishra, as said, and this is Joana, and she will start the talk, and I will join in halfway.
Yes, keeping separate. So good morning, everyone. As you said very nicely, we work in Kiel, which is like this tiny city in northern Germany. So Neha and I, and together with also Florian Tran, we work together in this project. You can find the project is open access in Immunity, and we also have some links on the end of the presentation that you can check, and you can also play, for example, with our single cells in the Fast Genomic platform and so on. So without further ado, I will start. Yeah, this was an endeavor that was not only done by us, by a lot of people in both Kiel and also in the hospital in Cologne. We also had a lot of the collaborators working together with the Lung Biological Network and also with the Deutsch COVID-19 Omics initiative done by DFG. So a lot of people contributed, and we just wanted to kind of send a shout out to all of them for helping us finishing this project in such a fast and timely manner. So just a little introduction. I think this is like everybody here is aware that COVID-19 is one disease. However, there are multiple disease courses that can occur. At the time that we started this project, there was kind of a shortening on the description of short and long-term disease outcomes for COVID-19. And still, even today, there is like no disease limiting medication available or just some trials, but they're not kind of set. We know now a little bit more about COVID-19. We know that there are differences, whether it is severe COVID-19 or mild COVID-19. So for example, once you have a severe COVID-19, you're more likely to have an increase in circulating neutrophils. There is a lot of studies that have described lymphopenia. Also, there is a reduced abundance of NK cells. And one of the things that also kind of relates back to our project is that there is a lot, like a big increase, significant of thrombotic complications. This is like 9 times more likely to occur than in diseases such as influenza. Also, one thing to take into account if you have, for example, mild COVID-19, is that there is an increase in the circulating inflammatory monocytes. So with our project, we wanted to go a little bit further and kind of in a smaller cohort, but really have an in-depth analysis at different omics level of COVID-19 and different types of the disease. So this is what we did. We had indeed a longitudinal sampling. So the sampling was done like every 2 weeks for 6 weeks, right? Sorry, it's been some time. And this sampling was done in 2 centers, so in the University Hospital of Kiel and Cologne. And not only we had different samples taken, we also had very dense clinical phenotyping. So we have what we call cohort 1 that was comprised of 14 patients. And we also have kind of a healthy control cohort. From this, we took samples of whole blood where we use bulk DNA and bulk RNA. So we did DNA methylation, bulk RNA-seq, and then also BCR-seq. And from the PBMCs, we did 5' single cell RNA-seq. And also we took some samples of serum where we did multiplex ELISAs, for example. Not only we had our own cohort collected in these 2 centers, but we also had validation cohorts. So the first validation cohort is a single cell cohort. And this is a cohort that's already been published in this Schulte-Schrepping paper. And then we have a cohort 3, which is what we call the Netherlands cohort, which is a cohort that has not been published as of now, but is a cohort that includes a lot of patients that have been sampling and done bulk RNA-seq analysis. So one of the novelties of our work is that instead of focusing on the patient within themselves, we try to have a better granularity, so a better characterization of the patient trajectory. This means that for the several samples taken from the same patient, we try to classify the samples themselves. We did this because it is indeed a heterogeneous disease, and also because when we say heterogeneity is not only in severity of the disease, but also in the speed and the disease course itself. So for the individual samples, we classify them based on the WHO ordinal scale, which is kind of like more or less how the ventilation of the patient at that moment of time. And we also use CRP, IL-6, and ferritin, which are known inflammatory markers, to classify the samples themselves. So then for each individual patient, we had what we call pseudotimes. Our pseudotimes go from 0 to 7, 0 being a healthy control. And then you have 1 and 2. So 1 is kind of when the patient is in an incremental stage of disease. So they're already hospitalized, but the disease is getting worse. Then we have critical, so 2 and 3, critical and complicated with hyperinflammation, which is like a very critical state of the disease. And unfortunately, sometimes the patient perish. And then we have the following pseudotimes, so 4, 5, and 6, which are more convalescence and also kind of mild disease pseudotimes. We also have some interesting points, which is, for example, here the 7, which is like a long-term follow-up of the same patients. So this is just for you to have an idea of the samples that we took. So here you can find principal component analysis. This is for the whole blood bulk RNA-seq data. So you can see here, for example, on the principal component 1 on the right side, there is in gray the healthy controls, which are kind of clustering, also together with the long-term follow-up, so the recovery patients. And then if you go further down the left, you could see that you find the more critical samples, and these are samples that are kind of in a darker color, so dark purple and so on. And this will be the same throughout the presentation. Also, something interesting here is that there were a lot of interspecific variability. However, for very critical states of the disease, you find that these samples cluster not with the patient, but more closely here on the left. So here is just an introduction to what we did at the single cell level. So we collected over 350,000 cells. The first thing that we did is like we took all these samples, took all the cells, and we did cell annotation. So here you can find the different cell types. Here on the left in blue, you find the monocytes, in orange CD4 T cells, this purple color CD8 T cells. Below in yellow, you can find B cells, and on top, for example, you find the megakaryocytes here in green. So once we colored the cells, not by the cell annotation, but by the pseudotimes, you can see already for very specific cell types, there is some type of clustering based on the pseudotimes themselves. So we were interested in these cell types that show this characteristic. So from now, I will focus on the megakaryocytes, which is here on the top, and you can already see there is some type of subclustering. So what I did is that I subset the megakaryocytes. There were almost 6,000 megakaryocytes in our cohort. And the first thing I did was to measure their proportion throughout the pseudotime. So here you can see that the very critical stages of the disease, there is kind of an increase in the number of megakaryocytes, and then it goes down. Also, not only there was different proportion of megakaryocytes at critical phases of the disease, but the megakaryocytes themselves were kind of clustering differently. So here you can see on the right, this is the UMAP analysis, and you can see this first UMAP on top, it corresponds to the healthy patients. And once you go down the pseudotimes, you could see there is kind of a reclustering, and they are not localizing any longer with the healthy megakaryocytes, meaning that the transcription profile of the megakaryocytes at the critical and hyperinflammatory phases is quite different than from the normal megakaryocytes of a healthy patient. So we wanted to have a closer look. One thing we also identify, unlikely what has been described before, but this is specific for the megakaryocytes, is that there is a prevalence on the type 1 interferon levels. So here you can find examples such as IFIT M3 and IFIT M2 that have very critical phases on the disease. This is kind of upregulated in relation to, for example, healthy or kind of more convalescent pseudotypes. The interesting thing is also that for the cohort 2, you can find the same pattern. So this is not specific to our project, but you can find it with other cohorts of severe COVID-19. Also, what we did then was to identify the differentially expressed genes and try to do gene ontology enrichment analysis. And as you can see here on the right, there was a prevalence on the enrichment of the cellular response of type 1 interferon. This is true for all the pseudotypes during COVID-19. And then in more critical phases of the disease, you find that the terms such as platelet degranulation, platelet aggregation are kind of enriched, especially in the critical stages. So I will now, Neha will continue.
Thank you, Joanna. And now I will continue with the analysis. So of course, single cell profiling has a big advantage that you can basically measure the transcriptome level at each individual cell, which you can't get with the bulk data. But it also has some drawbacks because some cell types, because of their size or because of the method that you're using, can be missed in single cell data, and the transcriptional signatures from these cell types will not be seen. For example, neutrophils have already been shown in several papers that granulocytes are very important in COVID-19, but in especially the method that we use, the 10x genomics method, we kind of miss most of them. So in parallel with the single cell profiling, we also did the bulk multi-omics profiling. And here I show you the two omics layers that we did. It was gene expression and DNA methylation. Now, in both the cases, we wanted to identify what genes or what sites, methylated sites, have changed in COVID as compared to healthy controls, as well as within the COVID with different disease trajectories or disease severities. So we did this in both of them. We compared the healthy controls to the COVID-19 patients, and we found some differentially expressed genes. And we also did a longitudinal analysis, and we combined them. And we did a similar thing for differentially methylated positions. So this is just a subset of the differentially expressed genes that we found that were very different across the different pseudotypes or across the disease trajectory. And here you can see, so the first one, the gray one, is the healthy, the 0 pseudotype, and the last one is the most critical one. And then the whole going up and down thing is in the middle. I put the critical in the end because many of these patients actually die, so they don't go down again. So here you can see some interesting patterns. For example, we see erythroid differentiation is very high in convalescent stage, but also in the critical stage, and I will talk about it also later. One interesting thing that many studies pointed out was the interferon 1 signaling, which they found that was kind of reduced in very critical disease. We also found the same thing. We found that it was high in inflammatory states in general, but when it comes to very critical, it was low. Then one other thing that was also shown in the Schulter-Sprepping paper with the granulocytes and the monocytes, that they found a lot of neutrophils in the severe COVID. And we also find this thing in the very critical stage. We have high transcripts that enrich in neutrophil degranulation or myeloid cell immunity. And then we also had, like, in general, the immune pathways like TLR signaling, interleukin 1 signaling, which was high in inflammatory stages, including the critical one. Then we, after we found the differentially methylated positions by analyzing the DNA methylation data, we wanted to integrate the DNA methylation and the gene expression because we wanted to know things at the transcript level, what's happening. So that for that, we had a very simple workflow. We just identified the differentially methylated positions that were in the 10 kb window of the differentially expressed genes. And then we did a simple correlation analysis to identify the genes that were potentially linked with DNA methylation. And then when we did the geoterm analysis for these genes, we find that many of the immune processes, so not only the housekeeping processes that you would expect, like the ATP metabolic process and so on, that could be really associated with DNA methylation, but also a lot of immune processes like interleukin 1 beta production, interleukin 8 secretion, interleukin 6 production was also linked with DNA methylation and increased in COVID-19. Now, going back to the cell types, what we wanted to also look at certain signatures from the bulk data that can be reflective of the individual cell types. And to do that, we did a co-expression analysis. So in this, you just identify the transcripts that have the similar expression pattern across the disease trajectories. And then once you have these modules, you cluster them based on the expression. And then once you have these modules, you can correlate with the cell type fraction, with clinical parameters, and just look at the expression patterns and so on. So using this, since Joana already introduced the megakaryocyte population, I will first focus on the modules that were related to the megakaryocyte cell proportions. So M3 and M4 in our data set were correlated with the cell type and also with D-dimers. And you can see here their expression of the transcripts from these modules were very high in the critical stage. And then it goes down, which is similar to what the megakaryocyte cell proportions look like from the single cell data. Then we wanted to see if whatever we observe here, is it of any clinical relevance? Can it be actually used in a clinical setting? And to do that, we used another cohort, which is the cohort 3, which Joana already introduced from Netherlands. This had 40 patients, and these were much more severely ill compared to also the cohort that we had. In these cases, the blood was taken from the patients from 2 time points after hospitalization. And then they did the bulk RNA-seq using the blood. Because some of the patients in this cohort unfortunately did not survive, we actually checked whether our transcriptomic signatures can predict or have some association with the mortality in this cohort. So, and we found that the M4 module, which is also linked with megakaryocytes, did have some association. So here, when you compare between the 2 time points from the same patients, in non-survivors, there was an increase in the transcripts of the M4 module, whereas not in the survivors. Then we focused on another gene expression module, gene co-expression module. So it's module M7. As I said before, I will talk about erythroid differentiation a little bit. So this module associated with the proportion of erythroid cells. And when you do the gene set enrichment analysis, you see that it's linked with hypoxia or heme regulation and so terms. The interesting thing of this module was it had a biphasic pattern. So you see it's high in the critical, then it goes down, but it's again high in the convalescent state. And the idea behind this could be, or our hypothesis is that it's very high in the critical, just showing whatever hypoxia happening at that point. But then also when the patients are weaned off the oxygen and their body is getting back to the normal supply of oxygen, you still again see that these transcripts are highly expressed. We could not identify too many erythroid cells from the single cell data because of their shape. But when we look at their progenitor cells, so MEPs, which are the progenitors of megakaryocytes and erythroid cells, we see, although not statistically significant, but we do see a biphasic pattern here. Then we did the same thing. We checked whether this erythroid cell signature was associated with mortality in the 3rd cohort, and we found that it was. So when we look at the M7 module, in non-survivors, there was an increase in expression of these genes, but not in the survivors. Also, when we looked at the genes that were different between the 2 time points and the non-survivors in general, and like doing a backward analysis, and then we map it back to our single cell data, we saw that many of them were from M7 modules, and also kind of mapped back to the whatever little population of erythroid cells we had. So we did kind of a multi-omics analysis for COVID to understand the COVID-19 trajectory. We tried to integrate the different layers as well as we could to get kind of a more holistic view of what's happening. And the take-home messages from the part of the study that we showed today is, first, we see an increased proportion of megakaryocytes, which sustains interferon 1 signature. And these signatures are actually correlated or associated with an unfavorable disease outcome. And the other cell type that I would like you to remember is the erythroid cell type. So there's emerging erythropoiesis, again, in the critical disease, which is also correlated with unfavorable disease outcome. And of course, there were so many people involved in this. But first, we would really, really like to thank the patients and their families for contributing to this study. We didn't have a big cohort because fortunately in Northern Germany at that time, not now anymore though. We didn't have that many cases, but whatever we had in the ICUs, in the hospitals in Kiel and Cologne, we are very grateful to the patients and their families for cooperating, and all the people who contributed, and especially the funding agencies. So this is a part of the DECOI initiative. It is the German COVID-19 omics initiative funded by the DFG, and also with the help of our University Excellence Cluster, which is the Precision Medicine and Chronic Inflammation. And you can check out our paper in immunity. We are also very happy if you use our data to do other stuff. You can also check out our codes on GitHub, and you can also email us if you, of course, you can't copy these links right now, but if you want, you can also email us and we will be happy to provide you. Thank you.
Thank you. Excellent work. Thank you so much for presenting. Does anyone have any question regarding the presentation? Or let me start with a question. It maybe seems stupid, but according to my understanding, cohort 2 and 3 were used for validations, and I'm wondering what's the purpose to use two different separate cohorts for validation instead of combining them together?
Yeah, so they were separate because cohort 2 was, we only had single cell sequencing data from cohort 2. We only had single cell RNA-seq. They did not do bulk sequencing in Bonn, where this cohort comes from. Whereas in the other cohort, we only had bulk sequencing data and no single cell. So it was mostly based on what kind of data we had. But we were also especially interested in cohort 3 because this was a combination of much more severe patients. Like the other cohort, the cohort 2 did have some severe patients, but most of it was mild and moderate. But the cohort 3 had much more severe patients, and we wanted to see how the signatures that we identify with a very small sample of severe patients that we had, and how they look in a larger cohort.
Okay,
and
thank
also
you.
cohort 2 also was different in their methodology for the single cell. So they had both. So cohort 2 was already a two-center cohort from Berlin and Bonn, and they had 10x data generated and also Rhapsody single cell data generated. So there was already a lot of like variability within. So we thought it would be like, instead of merging everything, also because if you work with single cell, you know, the more you put together, the more your computer dies. You know, so you also have to be careful. There's definitely that limitation.
Okay, I think Dr. Takahashi have questions.
Yeah, thank you for the very beautiful talk. It was really impressive. One obvious question from me is that, did you have chance to look at the sex difference in like gene trajectories or epigenetic trajectories? It would be very interesting thing to look at, but did you have chance to do that?
So no, we didn't do it because the sample size was very small. Mm-hmm. So if you do a sex difference at only 1 point, we only have 13 patients to do that. And you're right, especially for methylation, there could be big ones, big differences. And well, maybe if we recruit more patients for COVID and we do this methylation analysis, but even if we do transcriptomics, it would be worth looking at the sex differences.
Yeah, it would be very interesting. Yeah, thank you.
Any questions?
Thanks. I have a question regarding the pseudo time, because here you actually have actual time, right? So in my understanding, your pseudo time is actually a little different from the, you know, single cell analysis. Some of them use inferred pseudo time, but here your pseudo time is actually your longitudinal time points.
Yes.
Yeah. So
and do you see that actually you could have used that longitudinal time points to predict patients' severity if they only have data at one time point? You can, you know, guess by this time they're already almost done with the disease or early on, or are there indication you can do that with this kind of technique?
Yeah, I think that's an excellent point, and I think I have an example of one patient that we only had one time point that was included like that, taking into account everybody else. So the thing is with we started, of course, with the easiest way, which was the longitudinal time, but already there were so many differences because so we got the first sample upon hospitalization or just right after. And you could already see that some patients were coming from other clinics and were already severe, very severe. So and there were patients that had, for example, one of the patients had the disease for 12 weeks before it was even hospitalized. So you cannot really like take this patient and put it as time point zero. So that's why we try to kind of, you take advantage of what's already out there, which is the WHO. There are also the LEOS kind of scale, and then use the data that we have clinically available. So in our clinic, for example, IL-6 is always collected. So we already know a lot about these types of inflammatory because actually our basis is not on COVID-19, but we actually work in IBD. So we already know a little bit about what is the correct levels of inflammatory marker. So we took advantage of what we knew before, and we started to do like using the clinical parameters to kind of unsupervised clustering. And then we realized, even when we hand curated, that kind of the groups were grouping as we would if we were manually curating with the clinicians that are actually very involved in this type of diseases. So
I think you also meant whether our markers are able to predict by single time point. Did you?
yeah.
Yeah.
Yeah. So, well, we did see a trend when we looked at the Netherlands cohort. Like there was a trend of the difference when we were looked at the two modules that we checked, so M4 and M2. But the thing is, even in that case, they were taken at different stages of the disease. So like hospitalized for how many days, we don't know, and so on. Well, we know, but it's not the same. It's not the similar. So it's hard to say. And I guess your point is very important. That would be great if we could do it for like a single time point. But the idea behind, like, if a person is already in the hospital, they will probably be monitoring them, right, progressively. And so in that case, if we have a good marker that actually can tell you if this goes this direction, then your patient is going through a very is possible to go through a very unfavorable disease state, or so that was the idea behind it, and that's why we did it that way. But if we have a larger cohort and we have good enough markers, we can also try to check it for single time point.
Yeah, there's already some tries. So there's I know some people that are working with this type of clinical data, just putting more together and try to use this kind of pseudo analysis as well.
Thank you.
Okay, one of our audience, Huang, said a great work, and also asked, how can you freeze the whole blood down? Thank you.
Sorry, we normally are not the ones freezing the blood. So for the whole blood, we use PAX gene. And for the PBMCs, we use DMSO-based protocol. So you can find all of these little details in the manuscript. We are not the persons to ask for details in this aspect. We're so sorry. But
it's in the manuscript. You will find it there.
Yeah.